Okay, I think we can get started. Welcome, everyone. Thanks for joining us in this first installment of our 3-day workshop on managing orchards and vineyards for climate-friendly outcomes. My name is Melanie Jones, and I'm a professor in the biology department at UBC Okanagan, and I'm going to be your moderator today. As we begin, let me first acknowledge that UBC Okanagan is situated in the territory of the Silks Okanagan Nation and their peoples. I would also like to acknowledge that you're joining us today from many places, near and far, and we acknowledge the traditional owners and caretakers of those lands. So over the 3 days of this workshop, we're going to describe the results of a 5-year project funded by Agriculture and Agri-Food Canada through their agricultural greenhouse gases program. And our goal in this research was to study various aspects of orchard and vineyard management and how they affected soil organic matter and soil nitrogen. So the retention of those two and their loss. Our focus was mostly on irrigation, but as you'll see, especially on Wednesday and Thursday, we also investigated mulching a little bit as well. And there are two reasons that we focused on soil organic matter and soil nitrogen. The first one is the obvious one, that these are two really important components of healthy and productive agricultural soils. But the second reason is that when carbon and nitrogen are lost from soil to the atmosphere, they do so in the form of two greenhouse gases, carbon dioxide and nitrous oxide. So that's why we're interested in studying how to keep the carbon and the nitrogen in the soil and prevent their release to the atmosphere. So the focus of today's session is on soil organic matter and soil carbon. And we've got some really exciting results to share with you about soils in the Okanagan Valley. Tomorrow we'll report on the results on soil nitrogen. And on Thursday, there'll be two presentations looking at irrigation and mulch, more from a bird's eye view, looking at the environmental effects of those two practices as a component of the whole orchard and vineyard management. So it's a life cycle assessment approach. So your presenters today are my colleagues, Dr. Andrew Midwood, Uma Farhana Kushi, and David Emdy. Andy is a research scientist with a long interest in soil, plant interactions, biogeochemical cycling, and he's a world expert in the application of stable isotope approaches to studying those processes. He was responsible for a major survey of soils from vineyards and orchards in the Okanagan Valley. And I know some of you who are here today had some of your soils sampled by Andy. So that survey is the basis of a lot of the results we're going to show you today. Dave is a master's student at UBC Okanagan, and he's working on a project to collate and summarize the results of already published work on changes in soil organic carbon stocks associated with irrigated agriculture. And he's going to tell you about some sophisticated techniques that he used to look for consensus in those results on a global basis, and also some modeling that he's done. Kushi is a master's student also at UBC Okanagan, and she was working on a project to better understand soil inorganic carbon, and especially the contribution from the bicarbonates in irrigation water to greenhouse gas emissions. Okay, so just before we get started, there's a couple of housekeeping things. First of all, just so you know in advance, we're going to jump back and forth a few times between the three speakers just to keep things interesting. Their presentation should be about 45 minutes long. And then after that, we're going to have a Q&A session. So if you have questions for the panelists, you can enter them in the Q&A button. If you just look down at the bottom of your screen, you should see the Q&A button. And as we go through at any time, you can enter your question there. And the other thing you can do is if you go in there and you see a question already that's basically the same question as the one you have, you can upvote that question just by clicking on the thumbs up signal, and it'll move it up in priority. I also want to acknowledge the host of this session, this series today, which is BC Food Web. We're really thankful to the folks at BC Food Web for helping us organize this. BC Food Web is a web portal project that aims to increase access and connection to current research and other resource materials. And in addition, it aims to encourage innovation between farmers and researchers for future project collaborations. Okay, so now I'm going to hand it over to Andy to start things off.
Okay, thanks, Melanie. Hopefully you can all see my screen okay. So I thought I'd start out this webinar by just saying a little bit about soil organic matter. What is soil organic matter? Soil organic matter and the term soil organic carbon are often used interchangeably, and I think today as we go through this talk, we'll probably switch back and forward between the two terms actually, which and they basically soil organic matter can be thought of as is mainly composed of plant litter, so dead leaves, woody material and roots, and they're all in various stages of decay, from fragmented material right down to basically small compounds within the soil. And because of that, it makes soil organic matter quite difficult to characterize. And so instead of trying to do that, usually we just measure soil carbon, which is a lot easier to measure. And we tend to think of soil organic matter as basically being about 58% carbon. So carbon is is a big component of this organic matter we find in our soils, which kind of brings me on to what we call the carbon cycle. I just wanted to talk about this a little bit, just to kind of put it in context. So we've got soil organic matter here, which is all this litter and breakdown debris from dead plant material, essentially. But the whole process starts with photosynthesis, and this is where plants capture CO2 from the atmosphere and they, build plant tissue. And then on the other side of that is this process of soil organic matter turnover. So the bugs, the microbial community in the soil, bacteria, fungi, they are basically breaking down that litter all the time. And as they do that, they produce CO2 as well. So CO2 has been fixed and trapped out the atmosphere by photosynthesis, and it's been returned to the atmosphere by this whole community in the soil, if you like, and we call it soil respiration. So CO2 is going back in the other direction. And in the Okanagan Valley and in a lot of agricultural systems, we also have irrigation in the system too, and we're going to talk a little bit about how that impacts this carbon cycle. So CO2, we find a small amount of CO2 comes out of that irrigation water, and Kushi is going to talk about that later, so we'll talk about that. But essentially, what I want to kind of portray is this idea that we have this carbon in and carbon out all the time in the carbon cycle. So soils hold lots of carbon in this organic matter within the soil itself. And in fact, they hold 3 times the amount of carbon that we have in the living plants and the plants on the surface. 3 times as much of that is in the soil. So, there is a lot of carbon in there. And it's beneficial, it's hugely beneficial. It helps with nutrient cycling in the soil. It helps with water retention, biodiversity. So the sort of overall soil health is much better when we have higher amounts of organic matter. And it helps with structural quality, so the ability of soils to sort of resist compaction and things like that from tractors and general farm traffic is helped by a higher organic matter content. And all of those properties have a positive impact on crop yields as well. So it's, we see, soil organic matter generally is a good thing. We want to try and protect and increase it. Now, globally, if we kind of look at the global picture, there is a huge amount of organic matter in soils. So we've always said it's beneficial for all those reasons in terms of crop production. But one of the other things it's also, and what has brought more attention to it in recent times is the fact that we can lock away carbon in the soil, potentially for many years. And so there's a growing interest in, can we use soils to try and help reduce the amount of carbon dioxide in the atmosphere? And if we can increase the amount of organic matter, even by a small amount, that can have a very big impact. It's a kind of win-win scenario in that the plants benefit, crops benefit, but we also can draw down the CO2 from the atmosphere and help try and combat climate change, which is obviously a good thing. And I think particularly because of that last part, there's been a huge sort of upsurge in the research that's been going on now to really look at how we manage soil organic matter and how we can increase the amount of organic matter we have. So if we look globally, about 50 million square kilometers of soil are actively managed for food and livestock fiber production. And despite the fact that we say it's a good thing, we want to protect it, what we find, particularly with annual cropping systems, is the amount of organic matter is actually is being reduced. We lose organic matter in these systems. So when we take natural lands and we convert them into an agricultural system, we often see a very sharp decline in the amount of organic matter. So that's not a great thing, obviously. And it's been estimated that something like 116 petagrams of carbon, which is 116 trillion kilograms of carbon, has been lost from our soils. So, you're probably thinking 116 trillion kilograms, how on earth do you even sort of visualize a huge number like that? So I put this slide together just to try and kind of put that in some context. If you think about money, which is a good way of visualizing big numbers. So if you have a $100 bill, for example, a stack of 100 of those is $10,000. And if you had $1 billion, it would neatly fit on about 10 pallets. Kind of give you an idea just how much that is. But $1 trillion actually looks like this. So it's 10,000 pallets. So this is 50 pallets wide, 100 pallets long, and double stacked. And there's a little guy down there to give you sort of visualization of what a massive amount of a massive number that is. And we're talking about 116 trillion kilograms of soil of carbon being lost from agricultural soils. So bringing it back to soils, what does that? Just to kind of re-emphasize that point, in a handful of soil from this region, it's typically about 2% carbon. So if you had a kilogram of carbon, you need 2 metric tons of soil. So a really huge amount, and we've lost, as I said, 166 trillion kilograms of carbon. So massive amount of soil loss there. So soil organic matter is a valuable resource. We want to minimize the losses whenever we can, obviously, because it has these, this dual win-win purpose about it. And so we need to find ways to do that. And interesting, so the research, there's a lot of research around that subject now. And one of the areas of research that we focused on, and which surprisingly there's little known about, is how irrigation affects soil organic matter and irrigated agriculture in general. And so that was one of the first things in this project we wanted to sort of really tackle was, what is the role irrigation plays in the gain or loss of organic matter? So with that, I'm going to hand over to Dave now.
All right, so we know that soil organic matter is lost in agricultural soils compared to their natural plots. But the literature is also showing that there are some ways where we can try to bring that soil organic matter sort of up a little bit more, maybe not quite to where it was as a natural plot, but we can increase it. Management practices like reducing tillage, using cover crops, use of organic amendments like manures and compost, not removing crop residues from the plots as much as possible after harvest, and just generally increasing crop diversity, all increase carbon from year to year. And we know that irrigation as well is typically used alongside these practices, especially in climates like the Okanagan, where we're semi-arid and need irrigation to remain productive. We know that irrigation also tends to interact with these management practices, and that crops tend to be more productive because of it. In a lot of cases, the literature suggests that increases soil organic carbon, at least sort of near the surface of the soils. But more and more recently, that picture becomes less clear. There's recent studies that have shown that soil carbon decreases as you go down in depth into the soil, potentially even outweighing the increases right near the surface. And there's studies from New Zealand in managed agricultural pastures that actually show decreases at all depths of the soil, irrigated soils. So despite the fact that 40% of our food producing agricultural land across the globe relies on irrigation, there's not a lot of studies that actually look at just how those agricultural irrigated systems affect the carbon in those soils. So we wanted to take a look at, that. We want to take a look at seeing whether or not we can get a better understanding of how irrigated agricultural systems affect soil organic carbon. And to do that on a global scale, we used a technique called meta-analysis. That it's a method by which we collect the data sets and results from peer-reviewed published literature from around the world and gain new insights. So this is using materials that has already been peer-reviewed and has been shown to be scientifically sound, or combining the results of a number of these studies to take a look at, in our case, a slightly different question. The process goes a little something like this. So we start out by collecting the studies. We use online resources, journal databases, that's a kind of thing, with a very, very specific search term to try to screen out the studies that don't apply to what we're interested in looking at. Get rid of any duplicates, because you're bound to run across a couple of those. And then we need to screen the studies for quality and data. So take a look at the abstracts, take a look at the text in the articles. We need to then extract the data itself, get a big old spreadsheet and just start filling it with the data that we pull from these studies. Fill in gaps where possible. Sometimes we have to use rasters or other resources to fill in gaps where the information wasn't provided directly in the literature. And then we standardize the variables as much as possible so that they're all kind of talking the same language when we get the final data set. And then once we have that data set, we run new analysis on it. And in this specific case, we're looking at what happens to the soil organic carbon compared to natural sites. So we wanted to see adjacent natural sites and irrigated agriculture, if there's a difference in the carbon levels there. And what we found is represented here on the left. So this plot shows us the soil depth increments on the vertical axis and the change in carbon stocks as a percent on the horizontal axis here. The dotted gray line here is, for all intents and purposes, that's the natural plots. So any change either above or below that gray dotted line is the change represented in the soil organic carbon in the irrigated plots. So we can see that the soil organic carbon decreases at the surface by about 10%, which is a pretty significant amount when comparing irrigated plots to their natural adjacent plots. At the 15 to 30 centimeters, it's not quite so clear. The error bars here denote that there's probably not a significant change there, but we can still see the sort of trend in data. There is a bit of an increase likely. And then at the 30 centimeters and greater, there tends to be a bit of a decrease. But again, we're crossing that center line, so it may not be a significant, statistically significant change. But important one here is the overall. So what we're seeing is that when natural sites are compared to their irrigated counterparts, there is an overall loss of 5.4% carbon globally. But our studies have shown that maybe the Okanagan's a little different. These tend to be annual crops globally. The Okanagan has perennial crops, and I'm going to pass it back to Andy so he can talk about just how the Okanagan looks a little different.
Okay, thanks, Dave. Yeah, so irrigated woody perennial crops such as apples, cherries, and grapes are really quite different from annual crops for a number of reasons. One of them is that the individual plants themselves can grow for many years. In the Okanagan Valley, for example, the average age of apple trees is 22 years old. Cherries are 14 years old, and grapes on average are about 17 years old. And also, the soil beneath these crops is really undisturbed, sometimes for many, many years. So there's little or very little or no tillage of those soils. So they just can be undisturbed, as I say, for many years. And we have a kind of constant input of carbon from these trees. So there is constant input from the trees, but also in the drive rows between the crops, we have often grasses or weeds growing in there, which are also pushing carbon into the soil all the time. If you think about, that carbon cycle I talked about earlier. So there's this constant input, which is different from annual crops, because annual crops, there is always a period where a field is tilled and seeded, and then it's left fallow for a little while while the crops germinate. So there's, over that period, there's little input. And then obviously we have the fact that the crop is also irrigated over the growing season. So we were interested in, so how does that all work in the Okanagan Valley? So our big question was, how does agricultural production of irrigated woody perennial plants in the Okanagan Valley impact soil again? And also nitrogen. We're going to talk about nitrogen tomorrow in the seminar then, but today we're just focusing on carbon. So, spoiler alert, here's what we found, and it's a good news story actually. In the Okanagan Valley, irrigated agriculture has nearly doubled the amount of organic matter in the soils beneath those crops in comparison to native areas. So that's really great news, and it's kind of the opposite of this kind of global picture we see for annual crops. So perennial crops, irrigated perennial crops behave quite differently, and we see this big increase in carbon. So we're going to dig a little bit deeper into these results now. I'll just tell you how we got there and some of the other things we found along the way to this really nice conclusion of our work. So as Melanie mentioned, a big part of what we did was a soil survey. And this map on the side here in these dots is all the different sites that we visited over one summer collecting soils. And we went to apple, cherry, and apple and cherry orchards and vineyard sites, and also adjacent native sites, where, as far as we know, there'd been no agricultural activity ever. So they're just native areas, and we sampled them. We sampled almost 100 sites. We selected sites for specific soil properties. We tried to get a good geographic spread up and down the valley, and we also looked at 2 irrigation systems. We looked at drip and micro spray, not for all these crops, but for representative crops of the area. And we ended up with something like over 500 soil samples. So we had a very, really nice set of data to look at. So one of the first results that we found was not all of the soil organic matter is evenly distributed across a typical orchard or a vineyard. I'll just take you through this graph at the side here. So we've got this is the concentration of total organic carbon across the top, and then this is depth in the soil down the side. And you can see these blue circles represent the concentration, what we call the crop row. So this is that row right beneath the crop here. And then this is these black triangles represent the soil carbon concentrations in the drive rows. And you can see there's quite a difference in the concentration between the two, and much more carbon in the drive rows than in the crop rows, and it tends to decline with depth. So by about 30 to 60 centimeters, the difference disappears. But still, this is highly significant. So why is that? Well, we think grasses and weeds that should grow in these driveways are very effective at capturing CO2 from the atmosphere and producing, the turnover of the roots and fine root network produces lots of organic matter which pushes into the soil. So we think that's essentially what it is. These grasses and weeds are very good at picking up carbon and generating organic matter. The crops are at a disadvantage in some cases. I mean, many of them are grown on what we call dwarfing rootstocks, so they have smaller root systems, so they're not able to sort of push in the same amount of organic matter into the soil. But also where there's bare soil, obviously, there is, the restricted or reduced carbon input in that kind of where it's bare. And so that doesn't, that kind of limits the accumulation of organic matter there as well. So these are the reasons why we think we see these differences. So that was the first thing that we kind of discovered on our first kind of pass through the analysis as we were working on it. Now, not all the crops are the same. We found that it is influenced by the crop that is grown, and we found most organic matter is actually under cherry orchards. And we think that's really probably down to the fact these are large roots with relatively shallow, large trees with relatively shallow roots. You have a lot of litter input, a lot of leaves falling off these trees, and pruning debris is all going into the soils around these cherry trees. We found the least soil organic matter under vines. And here I think, they're generally vines are smaller plants and apples and cherries. And they have a different rooting system. They're quite deep roots, so they tend to be spraying the carbon that they are producing out into a larger soil volume, so we don't quite see the same sort of distribution of carbon. So that's probably the main reasons behind it. One of the other things we discovered, which was really interesting, is that and this is kind of well known. There are basically, you can think of organic matter is in two forms. There's two forms of organic matter. There's what we call this fast cycling labile organic matter. So this is organic matter which turns over quite quickly in the soil. It's maybe in the soil for a couple of years at the most, 2 or 3 years, before it's chewed up by the microbes and returned to the atmosphere as CO2. And then there's another fraction, which we call a stable or slow turnover carbon. And that can hang around in the soil for much, much longer, maybe hundreds or even thousands of years in some circumstances. So they behave very differently. And so what we found is as the amount of soil organic matter doubles, the relative ratio of those two types of organic matter stayed the same. So they both increased, but they didn't change in their relative ratios. Now the labile organic matter is interesting because this material that's turning over quickly, but this is what adds nutrients. It helps with nutrient cycling and the moisture retention and all those really positive things that I talked about earlier. So that that contributes to all of that. And the slower turnover carbon is interesting from another perspective, and it's interesting from this climate change mitigation, carbon sequestration perspective. And that is, the carbon that goes into that pool is locked away for a long time. And so that's, if you wanted to try and pull down CO2 from the atmosphere, that's the pool where we might want to focus our attention to try and, help combat climate change. And so we were interested to know just how big that pool is, and can we make it even bigger? And what is the capacity of the soils in this region to really be used to combat climate change? And so with that, I'm going to just turn it back over to Dave, who's going to talk a little bit more about that.
Okay, so there's a theoretical upper threshold to the amount of carbon that we can store in a soil. In particular with this slow turnover kind of carbon, because there's interactions between the carbon and the minerals. So there needs to be enough mineral to interact with that carbon. And that is the portion of carbon, that slow turnover, that we want to look at in terms of sequestering CO2 away for the long term. So that theoretical upper threshold, that we call sort of the upper limit, the difference between that and where the carbon stocks are currently sitting in a given soil is termed the carbon deficit. So which represents essentially the unused capacity to sequester additional carbon in that slow turnover pool. So my task was to determine if there is in fact room to put additional carbon in that slow turnover pool. So I used an approach that uses a lot of machine learning algorithms to determine what that upper limit is. But what it essentially boils down to is that upper limit is based on the samples gained from the soil samples. So it's based on real numbers, and what the machine learning models attempt to do is use what they call the 90th quantile of that data to determine the upper limit. And then we of course use the current carbon stock, and we just look at that difference. And what we found across the 7,804 hectares of orchards and vineyards in the Okanagan, of which the soil samples that we have are representative of, is that in the top soils, the 0 to 15 centimeters, there's 1.14 megagram carbon deficit per hectare, and 0.82 at the 15 to 30. So that's a pretty close to about 2 megagrams of carbon per hectare that we have the potential to store additionally in the slow carbon pool. And what that translates into over the Okanagan is this rather large 15,272 megagrams of carbon that we have the potential to store in that pool. That's about the equivalent of 6.6 million liters of gasoline. In terms of additional storage capacity. So that slow carbon pool, while it only represents about 30% of the overall carbon pool in our agricultural plots, still has a huge potential to sequester away carbon dioxide from the atmosphere to the tune of about 15.3 million kilograms in that top 30 centimeters. Now I'm going to hand it back to Andy to discuss just how we might look at using some of that unused storage capacity.
Okay, thanks, Dave. Yeah, so as I mentioned, I think what's very important to build up organic matter is that we have this continuous inputs of organic matter from plants growing in the system. And so whenever possible, we think, drive rows, for example, should have plant cover. We think, we need to think about how the crop rows, so the soil directly beneath the crop, is managed, and where I think we're possible trying to avoid bare soil there, if at all possible. So think about planting cover crops, for example, using organic amendments or mulches in that region there, and also adding maybe the drive row cuttings to the crop row along with pruning debris. All of those things can sort of help just to push more organic matter into the soil. So all of those things would be good things to do. When we think about building up organic matter and just doing any of this kind of work, we're always kind of conscious about this whole carbon cycle concept. So we're always thinking about, if we're accumulating organic matter, we've got to think of it in terms of the carbon that's coming in and the carbon that's also going out of the system all the time, and where are the roots for all those kind of carbon losses and gains that are happening. So I think when I talked about the carbon cycle earlier on, I mentioned this fact we have an irrigated agricultural system, and we found that there's a small amount of CO2 which comes out of this irrigation water. And Kushi is now just going to spend a little bit of time telling you about what the work we've done to better understand this this source of carbon. So with that, I'm going to hand over to Kushi now.
Thanks, Andy. A lot of effort is being spent understanding controls over the release of CO2 from soil organic carbon from agricultural land. but a very few works have been done demonstrating the impact of inorganic carbon stocks on carbon dynamics. So what is soil inorganic carbon? Soil inorganic carbon refers to the mineral forms of carbon in the soil, which consist mainly of carbonates. So it's a growing understanding that soil inorganic carbon can also be a source of CO2 from agricultural land. Recently, our team has found that if irrigation water containing calcium and bicarbonate ions is applied to the soil, then it can produce a small amount of CO2 from the soil, and it can also enrich the soil inorganic carbon pool. However, it was unclear how different soil properties affected the release of CO2 from irrigation water. So we investigated the impacts of a range of soil characteristics on soil CO2 emission from irrigation water. So for this experiment, soil samples were collected from different apple and cherry orchards, and also from some uncultivated areas across the Okanagan Valley up to a depth of 15 centimeters. And water samples were collected from Okanagan Lake that contains a significant amount of calcium and bicarbonate ions. So the collected samples were then taken to this laboratory. So in the laboratory, first some soil collars were prepared by cutting PVC pipe and fixing a fine nylon mesh and a plastic paper at the bottom of the soil collar to hold the soil inside the collar. Then the soil collars were packed with soil, but leaving some space on the top of the soil surface to allow the application of irrigation water. So then a Parpex chamber was secured to the top of the soil collar to capture the CO2 gas that was produced before and after the irrigation treatments. The gas sample was collected using a syringe and then injected into a Kynar bag that was connected to the Picardo ring down spectrophotometer which did the CO2 analysis. So after doing the analysis, we found that the amount of CO2 that was emitted from the bicarbonate in irrigation water, it was less than 10%. And it's actually an overestimation because the data were collected within few hours of irrigation. So it's a good news for us as the amount of CO2 derived from the bicarbonate carbon, it's very small. It's very small compared to the amount of CO2 captured by the living plants. So we can conclude that inorganic carbon from irrigation water is not a large source of CO2, and it can be outweighed by the CO2 taken up by the irrigated plants. So now I will get back to Andy. Andy will conclude our overall project.
Okay, thanks, Kishi. That was great. So yeah, just to conclude, we found that irrigated agriculture has a hugely beneficial effect on the soils of the Okanagan Valley, almost doubling the organic matter content, which is great because it promotes better crop yield, so nutrient cycling, all the things I talked about, healthier soils, improved structure. But we know, because we know the big proportion of that is fast cycling organic matter, we really need to, maintain the kind of continuous inputs to keep those levels at the levels they are now. And if things change and stop irrigating and change the management system, there's a good chance the amount of organic matter will slip back to the more natural level, so it would decline. So, so it makes you think, if we can just manage things in a particular way, we can probably maintain that high level organic matter, which is great. And as Kishi said, the carbon dioxide that is released from irrigation water really adds very little to the overall emissions, and is hugely outweighed by the carbon that's sequestered by plant growth and then subsequently deposited into the organic matter. So with that, I'm just going to bring our webinar to a close, I just wanted to mention that we have a fact sheet which summarizes some of these key findings, and that's available on the PC Food Network website. And here's a link for it here, and I think someone will drop that in the chat as well. And also, I just want to acknowledge all the orchardists and the vineyard managers out there who allowed us onto their properties, and I know some of them are joining us today, so we want to say thank you to you guys for letting us come on, take soil samples, answer all of our questions. Also want to acknowledge Kessin Hannam, Tom Forge, and Denise Nielsen from Agriculture Agri-Food Canada of the Summerland Research and Development Centre. They were really instrumental in getting this whole project going, and were really a big part of the overall team. And also we had a small army of students and research workers who helped. I won't read all the names there, but who helped collect the soils, process the soils, both from the university and from the Summerland Research Development Centre. And finally, we want to acknowledge the funding for this work, which was provided by Agriculture and Agri-Food Canada and the Agricultural Greenhouse Gases Programme. And so with that, I'll say thank you, and we'll move on to some questions.
Okay, great. Well, thank you so much to Andy and Kushi and Dave for sharing the results of their hard work over the last 3 to 5 years. And we'll move on to the questions. So again, I'll just encourage you to keep posting questions that come up, and they're getting forwarded on to me, the ones that are the most popular. So I'll just direct the questions then to our 3 presenters, and I'll maybe just identify one of them, but after that first person is given a go, then feel free, the other 2 of you, to jump in on the question. So the first one is the effective irrigation on soil organic carbon in non-alkaline soils is actually through increased productivity. So the question is, did you compare irrigation versus no irrigation under native vegetation? And I can quickly, even I can answer the question that's embedded there, which is, did we compare irrigation and no irrigation on native vegetation? We did not. But I thought maybe I'd give Dave a chance to address this, because he's the one that really did that meta-analysis, looking more broadly, so it would include those non-alkaline soils, comparing native and non-native vegetation, or sorry, the soil organic carbon underneath those. So Dave, would you be willing to tackle that first?
Sure, yeah. So yeah, the comparisons we did are, like Melanie said, not exactly what you're asking there. But I think the important thing about the comparison we did in the meta-analysis is that that conversion factor of native land to agriculture of various soil types. Because the biggest difference that we see, like we know that the SOC, this carbon in soils, decreases when we convert the native lands to agricultural, and that big disparity in carbon, in order to see increases in carbon at any depth, we have to cover that gap first, right? So any increases we see are actually quite substantial changes from the initiation of agriculture on those plots. The native vegetation is evidently going to be well adapted to that climate and those growing conditions, and is probably well suited to providing carbon to the soils. Yeah, I'm not quite sure what more to add there, but the fact that we saw overall sort of decreases between the native and the irrigated agricultural plots is a pretty notable thing, because a lot of the crops in the areas we're looking at in these semi-arid irrigated crops are annual. A lot of these food producing crops are annual crops, and in those situations we saw decreases when all depths were considered. I would be very interested to see that comparison of native irrigated plots versus non-native, or versus native non-irrigated plots. But we didn't look at those. That being said, actually, I did make mention to papers from New Zealand. There are pasture systems there that looked at the irrigated. I didn't show numbers there, but they have pasture systems in New Zealand where they irrigate the largely natural grasses and manage them as not as a crop, but for their cattle. But in those situations where their native grasses were under management, including irrigation, they saw carbon decreases. So we didn't look at them here in the Okanagan, but there are places in the world where they have looked at that and they still saw decreases. There's probably soil type variation in there that accounts for some of that, but that's
Great.
yeah.
Okay, thanks. Andy or Kushi, did you have anything to add to that?
I was just going to say, I think it's fair to recognize that in the native systems, it is the lack of water which really holds back those systems, and productivity in those systems is really restricted by a lack of water. So if you irrigated it, I'm sure you'd see some, quite different changes, but then would you, then it becomes, is it non-native then because you're adding artificial water amounts? So it's, swings and roundabouts, right?
Okay, great. Thanks. All right, the second question, let's come to the top of the list, is would you say that these results could apply to the Okanagan Valley soils as well? So maybe Andy, do you want to start off on that one?
Yes, and I would say yes, they would. I see no reason why not, is the short answer to that.
Mm-hmm. Well, and I guess the reason we could be fairly confident about that is that there were a really wide range of soil types that you sampled. I don't know if you want to elaborate on that.
Yeah, I mean, we deliberately selected sites which were representative of different soil types. So, for example, in the south, in Soy's region, they're very sandy soils typically, and then further north in places like Glenmore region, the Kelowna, very much more clay soil. So we deliberately selected right across these different soil textural ranges, different parent materials of the soils, and they would be very typical of soils within the small community, I think. So, yeah.
Great. Okay. Question number 3. Do different types of irrigation influence carbon capture? Does the amount of water absorption versus evaporation impact the results? So again, maybe Dave, I'll start with you, and then Andy might have some things to add in as well, because I know in your big meta-analysis you looked at different types of irrigation.
Yeah, so that's a great question. The short answer is yes, different types of irrigation influence carbon capture. My larger study, the results of which are global and don't necessarily directly apply to the Okanagan, but I looked at the effects of drip irrigation, sprinkler irrigation, and flood irrigation on soil organic carbon at various depths and whatnot, and they showed very, very different patterns. So drip irrigation tends to have very localized wetting, so you get a lot of you get increased crop productivity, but that the impact of the irrigation on the soil tends to be limited to directly under the drippers. So in a lot of cases, crop row or on the individual plants. And in a lot of those cases, what they found was that the impact on carbon in that soil didn't really extend past the wetted bulb under those drippers. So field-wide drip irrigation tended to show a decrease in carbon over time because those unirrigated portions of the soil were just, microbes are still doing their thing, albeit slower. So there's still CO2 efflux in those portions. Sprinkler tended to show the greatest increase overall. That even wetting of the soil with the more even percolation of water through the soil tended to sort of spread that increase in productivity to plant and non-plant species, or sorry, plant and crop and non-crop species alike. So some of those things that Andy saw with the drive row, natural vegetation increasing carbon also applies to regular other crops. And we tended to see an increase at most depths with sprinkler irrigation and flood irrigation, which we probably haven't seen the Okanagan here ever, or maybe for a very, very long time. Again, showed different patterns with increases at the surface, but not much below that, there's a lot more sort of changes in the soil characteristics in flooded soils. Nutrients and carbon and stuff get drawn through the soil much more quickly with that much water. Yeah.
And Andy, you have, I guess, a few results. The best comparison, I suppose, would be between apple with drip and apple with microspray. Did you find
right.
any?
We deliberately designed our sampling campaign to try and tease out some of these differences, but to be honest, we didn't see a difference in our data set. We looked for it, but we really didn't see any difference in the way the water was added. But I think that may be because we're doing. We visited 100 sites, and the way people irrigate, and there's all sorts of different management systems right across the board. So there's a lot of other factors in play, which makes it difficult to tease out just probably one.
Yeah, I think another important thing, especially given the direct comparison to what Andy and I just said, is that there's big crop differences, right? Andy's stuff is looking at perennial, orchards and vineyards, whereas the big global stuff I was just talking about includes a lot of annual crops. Probably mostly, it does include orchards and vineyards as well, but there's a lot of annual crops sort of mixed into that result.
Right. And so I don't know if either of you wanted, maybe Andy, the second question embedded here was, does the amount of water absorption versus evaporation impact the results?
Well, I guess it does. I mean, if you're losing the water that you're applying, that's obviously not a good thing. So yeah, if you can maximize that absorption, that would obviously be would be beneficial for sure. Yeah.
But I guess in terms of the carbon, I mean, micro sprinkler versus drip, you would think those two ratios would be quite different, but it wasn't enough to affect the carbon story.
No, not really. We couldn't pick it out. And we also found that the type of irrigation was also, in some instances, was also related to the type of soil. So in some areas, we found it very difficult to find drip irrigated apples, for example, where, the water is just, the soils are so coarse textured, it just drains away. So there's a lot of microspray in those systems.
Okay, here's next question was a good one for Kushi, I think. And the question is, what is the pH of irrigation water? We'll start with that one, and there's a follow-up. What's the pH of the irrigation water?
The pH of the irrigation water, it ranged between 7.5 to 8, or slightly above 8, 8.1 to like this.
And that was the Okanagan, the water from Okanagan Lake. Yeah. So presumably the pH of water that had come from other sources would be different. But Andy, I think in our study, most people were irrigating with Okanagan Lake water. Is that right?
I believe so. Yeah, mainly in the main. Yeah.
I think we were originally hoping that we could find some, enough sites so we could compare Okanagan Lake water with maybe some other sources that had a lower pH, not as alkaline, but I think I remember, Andy, it was a hard time finding enough of those.
I think so, and a lot of the irrigation water in this region just naturally has calcium carbonate in it, so much of a muchness really. We couldn't find any that were not rich in calcium carbonates.
Okay. And I don't know, maybe Kushi, I'll ask this follow-up to you too, but maybe Andy can answer it better. I'm not sure. The question was, how does carbon affect pH of the soil? Do you have any ideas about that? Or I know Andy looked at all of these data sheets, but you might have too when you were looking for matching soils.
It's a tricky question, actually. I would probably suggest that if we've got more organic matter in there, it tends to make the soil slightly more on the acidic side. But yeah, I really don't know the answer to that, to be honest, in this region.
Okay. So question 5, are there any potential harmful effects of this increase in organic matter in irrigated agricultural soils? Andy, do you want to tackle that?
Yeah,
Maybe it has to
I
do
would
with
say,
the different forms?
yeah, I don't know that there are, really. I don't know if anyone else wants to chip in, but I can't really think of any harmful effects. I think, you know, it's all to the positive. You can increase the organic matter of the soils. Yeah. We're starting from a fairly low baseline in soils here. And so, you know, sky's the limit, really. Yeah.
And it's going to be, you know, really hard. It would be really hard to have an enormous amount, because as Andy was saying, that most of it, 70%, is this labile fast turnover.
Yeah.
So, you know, it's never going to accumulate massively because it's being released, turned over and released all the time. So,
yeah. No,
Yeah, I mean,
there's any harmful effects.
organic matter at its root is plant matter, right? I mean, even if we're applying it as manure, it originated as plants. So plant matter in soils is all just part of the system.
And I mean, the places where you tend to see so much accumulation of organic matter, it tends to be in places where soils are really cold, you know, the environment is cold and that kind of thing. And so it's not decomposing. Whereas here, there's not really that issue.
Yeah, that's right. That's at the kind of extreme end of that is, you know, the kind of peaty soils where things are just all in suspended animation almost. So, but that's never going to be the case here. No. So no, it's all good.
Sorry.
No, I just said it's all good. Yeah.
Yeah. Okay. Next question, question 6. How does irrigated agriculture for orchards and vineyards change soil organic matter differently than other production systems, i.e. vegetables? So this is a good opportunity to kind of reiterate some of it's really a comparison of what Dave found and I guess what Andy found. So, you know, Dave's results that he presented was more from those annual kind of cropping systems, which would be like the vegetables. So I don't know if Dave, you want to give that a shot?
I can start off and let Andy fill in the gaps. The biggest thing it comes down to is differences in management practices on those types of fields, right? So the vineyards and orchards tend to be, I mean, while they're watched carefully and given amendments in irrigation and stuff like that, things like regular tillage or disruption of the soil doesn't really happen, especially not to the same degree as vegetables or other annual crops, right? And that disruption of the soil is really what reduces soil organic matter in those systems. So when you start churning up the soil, there's oxygen gets into the soil where it wasn't before. There's different water percolation characteristics and stuff. So basically the microbes are either, the microbial populations are different. They behave differently when the soil's been tilled. And in a lot of cases, what it means is that the microbes get access to oxygen and plant material that they can then turn into CO2 again, whereas normally it would remain a little bit more stable in the soil, as opposed to orchards and vineyards where they're not churned up regularly and given that oxygen and organic matter.
Yeah, I think that's true. And I think one of the other big, you know, key things which I mentioned is that this, you know, the amount of carbon going into the system, into the soil, is very different between orchards and vineyards. Basically, you've always got plant cover of some form and grasses in the dry rows and things like that. So they're always there and, you know, are turning over, you know, most of the year, except in the deepest parts of the winter. Whereas in vegetable systems, there are periods of the year where the ground is basically fallow and there's very little carbon input into the system. So that's the big difference, and that's why the perennial crops are so good at pushing carbon into the system.
And great, thank you. And then I think this is, unless I've misunderstood it, I think this is a similar question. I'm just skipping to number 8 just for a minute. The question was, do orchards and vineyards have a stronger carbon pull down potential than other agricultural systems? So if I understand that correctly, what our study showed was that it's the pull down part. I'm not quite sure if you mean pull down in the soil. What we had found was that that reduction in the soil carbon happens in the annual systems, annual cropping systems, but it did not happen in orchards and vineyards. So in orchards and vineyards, we're seeing a building up of soil organic carbon. So there's definitely a difference between those two systems.
Yeah.
Yeah. Okay, so let me then just go back to question 7. I think this is one for Andy. Did orchard size or age bring out some notable differences?
Oh, that's a good question. And I think, we've not really directly looked at this in the data, so we haven't statistically analyzed it, but my impression is that yes, it does. And I think because age means generally bigger plants, older plants. So particularly in some of the cherry orchards we sample, we saw some very old plants there, and they've been, very sites which have been undisturbed for a long time. So big trees, lots of litter and things like that. So I think that did. I'm not really sure about the size of the orchard. I don't know that would have had an impact, but definitely the age of the plants in there. And by age, I'm kind of translating that into then the size of the plants. We went to some apple orchards, which are very old trees and things like that. So yeah, but we didn't specifically analyze the data to look for that difference.
Okay. Dave Kushi, do you have anything else to add?
I know looking at annual crops globally, the age didn't really factor in, but that cycle, sort of the inputs versus outputs, is very different in those annual crops. So they didn't tend to accumulate the same way that the orchards and vineyards do.
Okay, great. So, so here's an interesting question. So if you are replanting cherry orchards from apple orchards, is it better to till it to as little depth as possible? I don't know. Andy, do you want to?
Thanks for that question, Nick. One of the people who kindly provided the site for us, or 2 sites actually. Yeah, I think if you want to preserve the carbon, yeah, I would say definitely do try not to till it too far, because I think any tillage on these soils is quite damaging. It really exposes that organic matter to loss. So if you can do it without tillage, deep tillage, I would say yes. Definitely.
Okay. And either of the other of you have anything to add to that, Kushi or Dave? Okay. All right. Next question. Why do vineyards have lower organic matter than cherry and apple orchards? What are some possible vineyard-specific solutions to increase organic matter? Andy, I think you've thought about
yeah, great. Yeah, so that's a great question actually, and I think there are a few things which make vineyards kind of not as good at pushing in organic matter as those other crops that we talked about. And I think just right off the bat, we're going to say that vineyards still increase the amount of organic matter in the soils relative to natives, relative to our native sites. But what I think one of the things is that vineyards tend to be irrigated less than cherries and apple orchards. So they, the amount of water in the systems is usually lower. So things are slightly more water stressed. So that kind of reduces the amount of organic matter being generated and turned over, I think, into the soils. The plants are generally a bit smaller. A vine is a smaller plant than a cherry tree, for example, but maybe not so much different from a high productivity apple orchard. But they have different rooting systems too. So the vines have very deep roots. So the root mass is kind of the roots are turning over amongst in a bigger volume of soil. So we maybe don't pick it up. I mean, maybe if we sampled deeper, we would see that more, but we didn't. So there's some of the reasons why we see those differences. Actually, how do we, possible vineyard-specific solutions to increase organic matter? I think certainly organic amendments to the crop rows would be a good one. I think how you treat the drive rows as well is something that I know there are some issues with that, where there is a worry that the drive row vegetation competes for water in the kind of water stress system with the vines. So there's some careful sort of management concerning that. But I think definitely don't till the derived rows if you can, and maybe use some organic amendments in the crop rows would all help build up organic matter in vineyards.
And I know some viticulturists are experimenting with cover crops under the vines as well. Yeah. And some of the scientists at the Agriculture Canada station in Summerland, they're following up on that too and studying the soil organic matter. So in a few years, we'll probably have a sense of whether or not that would be a good solution. Yeah. And I'll maybe just build on what Andy said too, that just as a general point, the roots really do contribute to soil organic matter quite a bit. And I think just as humans, because we live above ground, we tend to think of the prunings and the leaves and the above ground litter as what's really contributing to soil organic matter. But in other woody systems, like in forests, there's some really good studies that have shown that sometimes even more than 50% of the soil organic matter comes from the roots dying and then being decomposed and more roots being produced. So the whole rooting pattern that Andy referred to, that's quite important in terms of carbon.
Yeah.
Okay, next question. This is going to be Andy too, does having more labile organic matter than stable, so more labile than stable organic matter, increase greenhouse gas emissions? And if so, is there a way to decrease labile organic matter? So having the labile, does that produce more greenhouse gas emissions compared to stable?
Well, it doesn't really in the bigger scheme of things because it's this carbon cycle. So it's not, I think that's right. I'm saying it doesn't. It's a tricky question, that actually. I'm just I've kind of lost my train of thought on that. Can we have the question again? Sorry, I was just.
Sure. It's just, does having more labile than stable organic matter increase greenhouse gas emissions? And if so, is there a way to decrease the labile organic matter?
Yeah. Ooh. It's a really hard question to get my head around, actually, because that organic matter was originally in the atmosphere, it's trapped by plants, and it becomes the organic matter. I mean, I think I would say that we shouldn't look at the labile organic matter as being a bad thing. It's a tremendously good thing for plant growth, because it's the labile organic matter and it's that turnover of that material which releases nutrients for plant growth. So the labile is a great thing. So, really, I guess what you want to do is, it's always turning over. You can never stop it, but you'd want to increase, if you can increase that pool, that's a good thing. I mean, ultimately, I guess it is producing a little bit more CO2 than if you didn't have that pool, but there are other benefits, and you kind of hold it in the soil at least for a few years. And the stable, sorry, go on now.
No, I was going to say that's really, I mean, maybe the way is to flip it around and think about, is there a way to increase the stable?
Yeah, so that's the thing we were looking at with Dave's work, trying to increase that stable. The stable has a less of an impact on plant growth per se, because the carbon is kind of locked away, and it's locked away by kind of combining with the minerals in the soil. And so across this region, there is a limit to how much of that we can form because of the way the mineralogy of the soils is and the lack of weathering in our soils. But so the stable carbon is good for that, that kind of mitigation of climate change and locking away CO2 from the atmosphere. But it doesn't have so much of an effect on plant growth, I would say.
I think something else that's kind of important is, Andy, you showed, or you talked about that, the difference between the labile and the stable, but the proportions, right? That 70-30. It's all part of the same carbon pool. And we talk about them separately because they sort of mean different things to us, but by increasing the labile portion, you are also increasing the stable portion, because we saw that between native and irrigated plots, we increased overall carbon and that proportion stayed the same. So you are increasing one by increasing the other. So it's not a one or the other story.
No.
Okay, and so I think we're getting to the end. So the final question, so this is for all three of you. to give an answer. What was the most important finding of your work? So maybe Dave, I'll start with you, and then Kushi, and then Andy.
Right.
Sure. I think probably the most important thing, I mean, we saw that carbon in soils in the Okanagan, we're doing rather well, quite frankly, compared to globally, right? Globally, the stocks are down in irrigated agriculture in general, but we're doing well. We've sort of doubled our stocks compared to the natural plots. And that has a lot to do with a lot of different things. But just because we're doing well doesn't mean there isn't room to improve. And I think that's important to see too. Like that we saw that there is still room to grow in that bounded portion, in that slow turnover carbon stuff. So there is room to store more carbon in our soils, despite the fact that we're doing rather well when it comes to carbon storage in our soils.
Okay, thanks. Kushi, what about you? And you're muted, so.
So from my experiment, I found that the bicarbonate carbon in the Okanagan lake water, it releases CO2. I mean, it releases CO2, but compared to the irrigated crops, as irrigation almost double the plant production. I mean, crop production. And we know that crops capture CO2 from atmosphere. So, so when compared to the CO2 absorption by plants, that can double after irrigation. So this finding, I mean, the CO2 release from the bicarbonate in irrigation water, I think it's very small in amount. So yeah.
Great. But you were able to quantify that, which is great. Yeah. Okay. Thanks, Kushi. What about you, Andy?
I think really, the analysis we did on the labile and stable organic matter has been really revealing, because not only do we understand how the organic matter changes, but we also understand how it partitions between these two pools. And that's really opened up a whole range of things about how we can manage the soils differently, or the best way to manage the soils to increase the organic matter content. And I should say that interestingly, in the soils in this region, the relative ratio is quite different to lots of other soils that are out there. In lots of soils around the world, the biggest proportion is that stable fraction, and the labile fraction is generally a smaller proportion. That isn't the case in the soils of the Okanagan Valley. So that was really interesting, and it's something we've just published. Well, we're hoping to publish a paper on soon. It's with the review now.
Good.
That was good.
Okay, thanks all three of you again for a great presentation. Thanks very much. And yeah, this is perfect. Good. So you can see if you've got follow-up questions that you'd like to just follow up, separately with Andy or Kushi or Dave, there's their email addresses. And I'd also like to thank behind the scenes, you haven't seen them, but Jessica Lattis and Mairead Norton from BC Food Web. They're running this whole thing. They were collating the questions and feeding them to me. So thank you very much for that. And then just a reminder that tomorrow, same time, 11 to about 12:30, we have the second installment of the series, which is on nitrogen. So the microbial nitrogen cycle and contributions to nitrous oxide emissions. And then finally, feel free to visit the BC Food Web website, which you can see there. And we've got the fact sheet posted there, which is just a one-page simple summary of the major findings that you heard about today. So thank you all for coming, and hopefully we'll see you tomorrow.